This is supposed to represent molecules inside a bacterial cell. Membrane is up here. This is a flagellum. Bacterial flagella work a little weirdly compared to our flagella. This is DNA down here in the bottom, and all sorts of different proteins in the juice of the cell. It's not a great representation, is the molecules wouldn't be this big compared to the size of the cell, and they wouldn't probably be this crowded. They'd be jostling and moving around inside the juice, but there'd be some space around them for them to kind of bop into one another and move from place to place. But it gives you an idea of just how shape works into this. The membrane actually has two layers to it, because that's what you get with bacterial cells. And each of the layers has two layers of phospholipids. These would be lipid molecules with markers on the outside. So we're talking about proteins, and now we're going to get into what proteins do. So I'm going to back this up so I don't waste too much of this plastic that I'm writing on. Now this isn't even the whole list. There's too doggone many things that proteins do to give you the whole list. But when you think about shapes and the fact that you could produce virtually any shape by curling these things up inside and changing the face that faces the world, then there's a reason why we have protein-based chemistry, why our cells kind of run on proteins. A lot of structure in living things is associated with proteins. When we do the microscope lab and you look at your cheek cells are roundish / pentagonal. They're very flat. The nucleus is in a particular place inside the cells. Why do they all look like that? Well, because the proteins inside are hanging onto the membrane, hanging onto the nucleus in a particular spot, and giving them that particular shape. You could make a case for us being a particular shape because of the shape of our muscles, but that isn't really why our muscles are there. And a lot of our shape comes from our skeleton, which has some protein in it to give it some flexibility, but our skeletons are not really made of protein. We talked about how plant structure is carbohydrates, so structure for proteins is more on a cellular level, more on a little level, although our cartilage and our ligaments and our tendons are almost all protein. So there's some of our structure comes from protein. But structure on a cellular level, very, very, very much in animals comes from protein. When things actively move, you're looking at protein on some level or another. Our muscles, and I'll write this down in a second, our muscles are actually two proteins that have anchor points, and two of the proteins stick out from the anchors and the other one kind of rides in between, and they kind of ratchet past each other to pull millions of anchor points closer together and make your muscles shorter. Our muscles work by contracting, and when I contract this muscle, it stretches this muscle out so I can contract that one. And the contraction of the muscle is from proteins. I mentioned in the last segment that flagella and cilia, I didn't mention cilia, but flagella spin and cilia kind of push, and the movement comes from proteins. Microvilli that we'll talk about later wiggle from proteins. Our white blood cells crawl around using proteins to change their shape and to stick them down as they crawl. In our muscles, those two ratcheting proteins are called actin and myosin. Actin is an interesting molecule in that it's used for all sorts of different movement, including how our white blood cells crawl around or amoebas crawl around. And myosin is kind of a specialty protein that you find particularly in muscle. Myo is actually from the Latin word for muscle. But actin, from what I've read, things change over time, right? Evolution and all that. And even proteins will change an amino acid here, an amino acid there, which will change its shape. And as long as the overall shape of the protein stays the same and it can do what it's supposed to do, you don't get, you can get a whole bunch of different changes inside the protein. If you were to compare the primary, the order of amino acids in insulin from people to insulin from pigs, which they used to give people who were diabetic, it's still functional insulin. It still does what it's supposed to do, but the non-functional shapes are a little bit different. Some people couldn't take pig insulin. Their immune system was sensitive enough to go, this doesn't belong here, and they would actually reject it. Now we actually make human insulin using bacterial or yeast cells, and you're very unlikely to, it may be a very, very slightly different shape from your own personal insulin, but not enough for you to recognize it as foreign. So movement. Plant growth is actually dependent on a lot of protein interactions to make the plants grow. So even in plants, the movement, even though it's very slow movement, you can connect to proteins inside the plants. Yeah, they're adding carbohydrates to add to the structure, but the actual production of the growth is very much connected to proteins. A lot of molecules are involved in communications. When we talked about steroid hormones, I mentioned the hormones are produced in a couple of different types of molecules. Insulin is a protein-based hormone. Testosterone is a steroid, it's a lipid-based hormone. But we make, and animals and plants make a lot of protein-based hormones. They also communicate, plants and animals will both release chemicals into the environment that can be picked up by other individuals. The sexual signaling and other kinds of signals that are called pheromones. You kind of smell them. Now, in some cases, it's a signal that something bad is happening, and they call those alarmones. A plant that's being chewed on may release an alarmone that will both signal to plants in the neighborhoods and to other leaves on the same plant to maybe produce something that tastes bad, so that it's hard for the animal that's eating the plant to eat it all, because when it gets to the next leaf, it goes like, this doesn't taste so good. In some fiddleheads, it actually will make the leaves curl up. And they also will communicate with other plants that way. They also communicate through their roots through pheromones and alarmones. And there's other types of communication molecules that work like that signal from one place to another, from one individual to another. That's a U. Didn't start as a U, but it's supposed to be a U. In our nervous systems, our nerve cells carrying our signals are not directly connected to the next cell. There's a tiny gap, a very tiny gap, way tinier than this in reality. But they're trying to show. So the signal comes down the cell and hits this gap. And if a signal needs to start in the next cell, and here's where you can shut it off or redirect it, then what happens is proteins are released from one side, and they have to attach to enough receptors on the other side to start the signal back up on that side. Happens very, very quickly. Thousands of molecules go into the gap, go across this tiny little gap, and either connect on the other side or don't, depending on what's in there with them. You can release chemicals into the gap that keep them from going across. So our system has kind of switches in it so that we can control a lot of the function instead of it all being hardwired. But these are called neurotransmitters. They carry, and in some places like brains, it isn't just in the gaps. Neurotransmitters will actually activate a whole area of your brain. Like your pleasure centers are activated by neurotransmitters. They're kind of released into a zone. And these depend on receptors. Receptors pick up information. In the case of neurotransmitters, they pick up other molecules. In the case of hormones and pheromones, they pick up other molecules. But right now, if you're looking at this screen, you have receptors in your eyes that, that pick up various aspects of light. Sometimes light intensity, some of the receptors do that, some of them pick up different colors, some of them pick up different motion in different directions. So it isn't purely a receptor picking up a molecule, some of them respond to different types of energy. And so in communicating, they're the ones that kind of take the information and then do something to pass it along. Antibodies. Your immune system is always on the prowl, looking for molecules that are big enough to pick up. So little molecules kind of get by them. Big enough to pick up, but they don't belong to you. And so you have these patrolling white blood cells that will, they're like crawling over your surfaces, and if a bacterium from outside bumps into them, they attach to it and go, wait a minute, this molecule sticking out of the bacterium doesn't match the US list. This molecule sticking out of this virus doesn't fit the US list. And what the cell does is it literally rips the molecule off the surface and hands it to another cell in a particular way and says, okay, we need to be able to react to this. Say you pick up the coronavirus. You have a kind of a generalized fighting virus system that will do a pretty decent job in most people of keeping the coronavirus from getting to the point where it really harms or kills you. And because you need to have that, because this system, this antibody system, doesn't kick into place that quickly. It takes a while to make antibodies. It may take days, it may take weeks in some cases. But when you finally have an antibody made, say you've kind of gotten over the coronavirus on your own, with your own upfront thing. Now you're making antibodies. And this end has been particularly made to attach to the molecule that sticks outside of the coronavirus, or that sticks out of the bacterium you've been exposed to, or whatever you've been exposed to. This molecule that you're reacting to is called an antigen. It generates an anti-response. And now you have a specific way to fight that specific invader. Like, I had COVID. I was checked for antibodies. Having had the actual virus, I have antibodies to probably a whole bunch of different molecules sticking out of the virus. So it's a very kind of broad-based response. I'm going to shut the window because it's getting noisy. And that's how your immune system keeps you from getting sick again. You get exposed to the virus a second time. Your antibodies attach to the antigens. The other end of the molecule is an activation signal. If this changes shape because it's attached to something, this changes shape as a signal to your immune system that goes, hey, guys, a whole bunch of different types of cells react. Come in, and whatever I'm attached to, deal with it. And they may come in and gobble it up and digest it, or they may throw oxygen radicals at it to make it fall apart. This is why antioxidants are not necessarily the best thing in the world to be taken a lot of. There's a whole bunch of different ways that what they call the killer cells will come in and go after the virus or the bacteria, whatever the invader is. So it's a really important protein, but it's kind of amazing that we kind of make this stuff on demand to fit a whole slew of molecules we haven't even been exposed to. This is what they do with vaccines. With a vaccine, the idea is we don't need to expose you to the real virus that can make you sick. We just need to expose you to the right antigens. And then you'll make antibodies before you ever get exposed to the virus, but they'll be ready to attach to the virus, flag it, and it'll never set up shop in you. With the coronavirus, like right now I was sick in March. It is now September. if you check my antibody levels against what I had in March, they're probably lower because I don't have any viruses to fight. So I'm not cranking out huge amounts of antibodies all the time. I can if I get exposed again. And based upon that drop in antibodies, people are going, we're not really sure you're immune for very long. But like we talked about in experimental design, antibody measurement is not a great measurement for how ready your immune system is to react to something. But looking for those particular cells that are coronavirus particular cells, you've got cells ready to crank stuff out, but isolating and testing and counting those is very, very difficult. So they use the antibody levels. When they're running the vaccine tests, they're looking for antibodies, but they are actually going to the expense of looking for those particular cells, because those cells are really going to tell you if you're responding in a way that will keep the virus from attacking you. Normally I wouldn't go into that much detail, but we're in the middle of a pandemic, so it's kind of important. Still on the list of what proteins do. Enzymes. Enzymes speed up chemical reactions. Most of the stuff that happens in your cells, if you put it into a test tube, it's not going to happen. Maybe if you heated it way up, but you can't do that in your cells because the molecules don't hit each other the right way very often, or they don't spontaneously break apart very often. What an enzyme will do is it's sticky for the molecules that need to work. And so it has what's called an active site. You take in cane sugar, sucrose. There's an enzyme in your digestive system that has a dent in it that is the shape of sucrose. Sucrose fits in it, and when sucrose sits in that active site, the enzyme changes shape a little bit and stresses the hell out of the molecule's connection point, holding the fructose and the glucose together. And they break apart way more easily than they would if you just waited for them to break apart. And in some cases, like you need to build glycogen, that starch. Inside your liver cells, you have an enzyme with a place for glucose where it can be attached to another glucose, and then an open site for new glucose. And as glucose is pumped into your cells, it fits into the first molecule, or the first active site. The enzyme changes shape. It goes doink, and it connects that glucose to the string that it's already been building. It kind of moves the glucose over, so that now what's in the first dent is the glucose on the string, and now it's got a dent ready to grab another glucose. So it's building stuff. I know I went fast, but the idea is it can grab things and slam them together to help them build. It can grab things and stress them to get them to break apart. That's an awful lot of the chemistry that happens in your cells. It can move stuff from place to place. Enzymes, our protein-based chemistry is mostly enzymes. And except for ones that are really old and were named before the rule came along, if you see something with an ASE ending, it is an enzyme. It is one of the rules that enzymes are named with ASE endings. Okay, I don't know if this is a use, but I'm putting it on the end of this list. And as I said, this is not the whole list of uses, but these are kind of the big hitters here. Now, I don't know that my daughter is ever going to forgive me for explaining to her biologically why zombies just can't work. Is that the rules for zombies just biologically just don't, they wouldn't function properly. So we don't need to worry about the zombie apocalypse. And there is one place in the world where there are zombies, and that's this. Prions are a very bizarre type of protein. Their ability is to attach to a protein that matches it on its primary sequence. So it's the same string, but a different shape. And the prion can attach to that and fiddle with it, and when it comes loose, it has changed shape to match the prion. So it's like being bitten by a zombie and then being a zombie yourself. So the prion comes in, attaches to a protein that is like it but not like it, and fiddles with it until it comes off, and now there's two prions. And they can attach to that same protein. Now the problem is, this prion gets into your cells, hooks onto this protein. That protein was doing something. You change it into a prion, it isn't doing that function anymore. And so it can gradually shut down whatever the protein was actually doing. And so prions can cause disease that way. They get into the cells, and as they turn a functioning protein into prions, the function gets less and less. I mean, you start to replace them. And in some prion diseases, it can take years and years and years and years to kind of get far enough ahead of the function that you start to get symptoms. There's some what they call Creutzfeldt-Jakob disease that some kids picked up from the prion from hormone preparations, where they squished the growth hormone out of pituitary glands, not realizing they were also squishing out prions from some contaminated sources. And they would give the kids growth hormone injections who were deficient in growth hormones. And the prions would get into their brains and cause this debilitating disease, Creutzfeldt-Jakob syndrome, but it would take like 30 years. Mad cow disease is a variant of that acts much, much faster. But it also gets into your brain cells, takes a working protein, changes it into prions, you lose the function, your brain stops functioning after a while. Worse if you're a cow. Okay, that is the list of protein uses, so that's it for proteins. And now we've got one more type of molecule to count for this particular chapter, and we'll do that in the last video or two videos. I'm not sure which it'll work.